Dear viewers, greetings. In this video, we are going to see about the 6 types of atomic absorption spectroscopy. The atomic absorption spectroscopy is a technique used to analyze the concentration of elements in a sample by measuring the absorption of light. Different types of atomic absorption spectroscopy are based on the method used for atomization and detection. Here there are 6 main types. They are flame atomic absorption spectroscopy or FAAS, graphite furnace atomic absorption spectroscopy or GFAAS, hydride generation atomic absorption spectroscopy or HGAAS, cold vapor atomic absorption spectroscopy or CVAAS, Zeeman effect atomic absorption spectroscopy or ZEASS, and finally laser enhanced atomic absorption spectroscopy or LEAAS. The first type of atomic absorption spectroscopy is flame atomic absorption spectroscopy. Principle: in flame atomic absorption spectroscopy, the sample is aspired into a flame where it is atomized and the absorption of light by the free atoms in the flame is measured. Advantages: this technique is simple, cost effective and widely used for analyzing trace metals, and this technique is suitable for elements like sodium, potassium, calcium, etc. Limitations: lower sensitivity when compared to other techniques, and this technique is not suitable for volatile elements. The second type is graphite furnace atomic absorption spectroscopy. Working principle: in graphite furnace atomic absorption spectroscopy, which is also called as electrothermal atomic absorption spectroscopy, the sample is placed in a small graphite tube which is then heated to very high temperatures to atomize the sample. Advantages: GFAAS has higher sensitivity when compared to the flame atomic absorption spectroscopy and suitable for trace level analysis, and it requires small sample volumes and applicable to a wide range of elements. Limitations: this graphite furnace atomic absorption spectroscopy is more complex in nature because it needs expensive equipments, and it is slower than flame atomic absorption spectroscopy due to the heating and cooling cycles. The third type of atomic absorption spectroscopy is hydride generation atomic absorption spectroscopy. In short form, it is called as HGAAS. Working principle: in hydride generation atomic absorption spectroscopy, volatile hydrides of certain elements such as arsenic, antimony and selenium are generated in a chemical reaction and transported to the atomizer where the absorption is measured. Advantages: this technique is a very high sensitive technique for elements that form volatile hydrides, and specially technique for elements like arsenic, selenium and tellurium. Regarding the limitations, this technique is limited to specific element that can form hydrides. The fourth type of atomic absorption spectroscopy is cold vapor atomic absorption spectroscopy. In short form, it is called as CVAAS. Principle: the cold vapor atomic absorption spectroscopy is specific to mercury analysis. The sample is chemically reduced to mercury vapor, which is then introduced into a atomizer for detection. Advantages: this technique is extremely sensitive and specific for mercury detection, and limitations: this technique is applicable only to the mercury. The fifth type of atomic absorption spectroscopy is ZEAAS. Principle: the Zeeman effect atomic absorption spectroscopy technique uses a magnetic field to split the atomic absorption lines, that is called as Zeeman effect, and reducing interference in the signal and improving accuracy. Advantages: this technique is used to correct for background absorption in high matrix samples and provides greater precision in the complex samples. Regarding the limitations, this technique is more expensive than the standard atomic absorption spectroscopy. The sixth type of atomic absorption spectroscopy is laser enhanced atomic absorption spectroscopy. In short form, it is called as LEAAS. Principle: the laser enhanced atomic absorption spectroscopy enhances the atomic absorption signal by using a laser source for excitation, improving sensitivity and selectivity. Advantages: this technique allows highly selective and sensitive measurements, and this technique is suitable for trace element analysis in complex matrices. Regarding the limitations, the laser enhanced atomic absorption spectroscopy is very expensive and it needs complex setup. Dear viewers, that's all about the types of atomic absorption spectroscopy. Thank you for your support. Thank you.